Okay, welcome back, everyone. Hope you had a good short break there. We'll resume the session with 4 more talks. We've got Dian Latifah, Jai Kai, Andrew Crawford, and Ronnie Irawanto in this session. The first one is by Dian, seed conservation and utilisation in Indonesian botanic gardens, seed banks. Then we've got, from Jai, we've got seed banking, China's plant diversity. Andrew's going to talk about achievements of 25 years of conservation of seed banking in WA. And then the final talk from Ronnie is a seed exploration and collection of wetland riparian seed in East Java. So, Dian, please start your presentation now. Thank you.
You're welcome. Thank you. Hello, everyone. I'm Dian from Seed Bank of Research Center for Plant Conservation, Indonesian Institute of Sciences, or now National Agency for Research and Innovation, or BRIN. I'm going to present seed conservation and utilization in Indonesian botanic gardens seed banks. The species-rich Indonesian flora is facing many threats, such as widespread deforestation for oil palm and timber plantations, fire, mining, natural disasters, as well as climate change. Therefore, various ex situ conservation strategies have been implemented to protect Indonesia's plant genetic resources, including the establishment and development of a national seed bank for wild plant species at Research Center for Plant Conservation, or RCPC, National Agency for Research and Innovation, previously Lembaga Ilmu Pengetahuan Indonesia, Indonesian Institute of Sciences, or LIPI, based in Bogor Botanic Gardens. We chose seed banking as one of our ex situ conservation strategies, so we have to be trained and be part of international community for seed conservation, focusing on wild plant species. We collaborate with Millennium Seed Bank Royal Botanic Gardens, Kew, UK. Then we implement it as our seed banking protocols. Bogor Botanic Gardens has been mandated to develop and supervise 43 botanic gardens in Indonesia, consisting of 5 national botanic gardens under National Agency for Research and Innovation, 3 botanic gardens under universities, and 35 botanic gardens managed by local governments. This is potential to collaborate to establish a small seed bank facility, or we call it mini seed bank. They can conserve their native and duplicate their seed collections in Bogor Botanic Gardens seed bank. So far, there are seed banks at 4 these botanic gardens, including the RCPC seed bank at Bogor, with 3 more at Cibodas, in West Java or Java Island, Prodadi in East Java, and Eka Karya Bali Botanic Gardens in Bali Island, all of which send duplicates of selected seed collections to the main RCPC seed bank. Together, these 4 seed banks hold 1,237 accessions covering 994 species, including 442 native species, including as well 133 orchid species that was counted in. It is estimated that there are around 30,000 seed-bearing plants in Indonesia. So the botanic garden seed banks have so far only served around 3.3% of the national flora. There is clearly much more work to be done. Regarding the database management, we are ongoing process to integrate our database with data warehouse of Millennium Seed Bank. The RCPC Seed Bank has made good use of its collections, utilizing 3,970 seed collections between 2014 until 2019 for research, academic purposes, and revegetation. As a seed bank located in tropical areas, we also implement some protocols to conserve exceptional species, such as the application of cryopreservation. We are also open to expand collaboration with Australian seed bank partnerships. Australia was my place where I pursued my postgraduate degrees at James Cook University, Tonsil, 10 years ago. This ends of my presentation. I'd welcome for any comments and questions. Thank you.
Hello, everyone. This is Jay from China. I'm from Germplasm Bank of Wild Species based at Kunming Botany, Chinese Academy of Sciences. So I'll give you a brief about what we have done in the past 15 or 16 years in China in terms of the seed conservation. As most of you know, China has a mega biodiversity. We have nearly 30,000 plants and lots of unique animals, and also China was the host at least the 4 biodiversity hotspots of the world. And then what you can see, the orange triangle was where the China Seed Bank located at. It's called Kunming. So as a party of the Convention on Biological Diversity, China also thinking of how to preserve the unique biodiversity in the early this century. So at 1999, a project called Germplasm Bank of Wild Species was conceived and was granted at 2005. So this project was thinking of to preserve China's unique wild species. So which are including 5 different kind of banks, including seed bank, plant tissue bank, plant DNA bank, and animal and microbians also be considered into this program. So what I'm going to talk about the seed bank is, I will spend most of the time talking about the seed bank, what I have done. So we are lucky enough to, at that time, we are lucky enough to get involved with Tues Global Seed Conservation Initiative at 2004. So we signed 2 agreements with Q since 2004 until 2024. So from the techniques and the trainings and the research programs, we all received the helps and from Kew Gardens. All these techniques was, of course, the one of the most important thing was to do the seed banking or to do the seed collecting of China. So all these techniques we got was cascaded to China's different partners. So in the past nearly 15 years, we did more than 115 collecting partners across China. And you can see this is the locality we have the seed collection. Come from. And the northwest and the southwest was the main area where we did make the most collecting program. So far, by the end of last year, we already had more than 10,000 species of angiosperm seed plants was conserved, which was about 36% of China's seed flora, about 85,000 accessions. We also received some international duplications, mainly from Kew Gardens and the World Agricultural Center, as a backup to safeguard the seed collections from other regions. So just give you a very quick conservation value of our seed collections. At the family level, we had China's more than 85% of families was preserved. and about 2/3 genus was preserved as seeds in a seed bank, and also 1/3 of the species was of China's flora. So have a very detailed about analysis of these seed collections. Yeah, we are doing well. Well, we are doing okay in terms of the threatened species, because most of the China's threatened species are potentially produce recalcitrant or intermediate seeds, or have very small, a very small population, limited individuals. So it's difficult to get enough seeds. And we got about more than a quarter China's endemic species was preserved, which over 4,000 species. And in terms of the medicinal plants, we are medicinal plants was one of the most important part we are focusing on. So now we have nearly half of the medicinal plant, or half of the plant species which have the medicinal properties was preserved in the bank. In terms of the CWR, we are China has not much CWR species, but we still try our best to preserve them. And actually, some of them are quite difficult in terms of the taxonomy. Most of the grass families. So recently, we are the evolutionary diversity also was think of very important for conservation. So this is a tree of life. This is the tree of life of seed plants at the family level. So we already got. Yeah, we are doing okay at this moment. So still a lot of families were to be collected from our countries. Actually, some of them are very tricky. We still don't know whether they are bankable or not. So still a lot of work needs to be done before we do the seed conservation on these families. In terms of the data and the collection size, most of the collections we have the plant name, we have the ID, and have the georeference data. And of course, we are one thing we are doing is we are also we collecting seeds, herbarium vouchers, status, and we're also collecting associated DNA samples for the potentially use the DNA for barcoding project. And in terms of this collection size, nearly half of the collections we have a large quantity of each sessions. So we try to maximize the quantities of each sessions. And for some with very limited seeds, we just grow them or just leave them in the field. And we also do the germination test. So for 60% of the germination, 60% of the tested collections will have the more than 75% germination rates. And every year we do the regular germination test, and now nearly 90% of the collections have started the germination test. And we also started the germination retest about 3 years ago, 5 years ago. So also accumulated some data, but about 11% of the collections have their viability was a little bit dropped. And we also have the try to better use of the collections. We also developed the web-based application system. So for for the Chinese user, they can access to this website and to access to the just do the online shopping, like use the shopping list and to order what they have, what they want. And so we could produce all these requests. So far, we had more than 8,000 accessions was distributed. More than that, should be more than 10,000 accessions were distributed. But our end user was mainly from research institutes or universities. Was mainly based at for the academic research. And about 2 years ago, the country was also encouraged to form a nationwide plant germplasm resources center. It's more like a wide germplasm resource network. So we are, as the main hub, to leading other at national or regional participants to put up their resources and their facilities together to work together to preserve the plant germplasm. But the main part for us was still based at seeds. So of course we also had a very good social impact. Also, it's also very important resources to get in touch with, get involved with the publics. In terms of the future, we are still thinking of to optimize our sampling strategy, especially how to maximize the genetic diversity for the species based on the research, based on the recent research. And we also think of to do the germplasm bank of wild species, the second, to have a new phase, to have a broader topic, including the macro fungi to do, and also the quality control was also another issue, especially when we were also thinking about when the viability was dropped down, how to improve them. And of course, we are also seeking the international collaborations, especially for the transboundary works in Indo-Burma and the Himalaya hotspots. And I also take this opportunity to thanks Kew Gardens to help our seed bank from the starting point and now we are the same as fully developed, but we still try to keep close with Kew, working on the global seed conservation. And thank you for your listening.
Firstly, I'd like to acknowledge the Whadjuk people as the traditional owners of the lands and waters on which I'm talking to you today. My name is Andrew Crawford and I manage the West Australian Seed Centre in Kensington. Today I'm going to be talking to you about the work the Centre has done over the nearly 3 decades of its existence. In 1992, a conservation seed bank, the Threatened Flora Seed Centre, was established in Western Australia. The centre was established to conserve species that were being threatened by the plant disease Phytophthora cinnamomi. Although humble in appearance, being housed in transportable buildings, the centre employed conditions aimed at storing seed long-term, that is, drying seed to low moisture content and then freezing the seed. Then, in 2010, the centre moved to a purpose-built facility that forms part of the Kieran McNamara Conservation Science Centre and is co-located with the West Australian Herbarium. The seed bank now forms part of the West Australian Seed Centre and is the primary repository for seed of all Western Australia's conservation significant plant species. When the seed bank first started, there were over 1,600 plant species in Western Australia of conservation significance. 270 of these were recognised as threatened species, and over 1,400 were considered priority species. These are plants that are poorly known, likely to be threatened, and in need of further survey. The number of conservation significant species has now doubled, with over 400 threatened plant species and over 3,000 priority species. On a good note, the number of species listed as being presumed extinct has gone down. So how successful have we been at collecting these species? The centre currently holds over 6,000 accessions, representing over 1,900 species. Over 2,500 of these are collections of threatened species, representing 346 species, or 81% of the species currently listed as threatened in Western Australia. Over 1,200 collections, representing 743 species, are collections of priority species, representing 23% of the current species listed as priority in Western Australia. The remainder of the collection is made up of common species. So we've done a good job of collecting the state's threatened plant species, and we've started to make inroads in preserving the state's priority plant species. But as most of the species being targeted are threatened and often low in numbers, the size of the collections is often small. For our threatened species, 3/4 of the collections are composed of fewer than 1,000 seed, with nearly a third of the collections being less than 100 seed in size. This poses many challenges when it comes to determining the viability of collections and monitoring their viability through time. The aim of the Centre isn't just to have a sample of each of our threatened species, but to hold collections that ideally represent the diversity of the species in the wild and to have sufficient quantities of seed available to undertake recovery actions such as translocation. Therefore, multiple collections are made of each species to achieve each of these goals. On average, for our threatened species, there's close to 8 collections for each, with some represented by up to 72 collections. If we look at the total number of seed in storage for a species, only around a third are represented by an amount that might be considered adequate, that is, over 10,000 seed, whilst another third of collections total less than 1,000 seed. So we've got all these collections that we've been storing for close to 30 years, and we want them to be viable when we come to use them. So, how has their viability been going whilst they've been in storage? So here is a snapshot of the performance of some of our collections from a project that looked at the viability of 44 collections representing 22 species that have been in storage for at least 25 years. Overall, most of the collections hadn't shown any significant decline in germination. There were 3 collections that declined in germination, all representing different species and genera. Whilst any decline in viability is of concern, for one of the species there was a collection that had been tested that had been in storage for a similar length of time, where the viability was still as good as when it had been stored. So, at least in this instance, it appears that collection quality may be an issue rather than longevity of the species. One of the problems we do face with using germination as an indication of viability is that the treatments that we use initially may not have been optimal and therefore don't give a true reflection of the viability of the collection. For the sample of collections I've presented, there were 4 collections, each of a different species, where the germination result in the retest was much higher than the initial test. The problem here is that we don't know how these collections have performed over time, because the initial viability may have been higher again and has dropped to the current value, or it may have stayed the same. I spoke earlier about how we make multiple collections aiming to represent the diversity of a species. I'm going to give you a brief example about why this can be so important. The Wongan trigger plant, Stelidium chroniforme, was originally known from a few populations in the Wongan Hills area. In the late 80s, it was discovered over 100 kilometres to the north of the known populations. Then, in the early 90s, seed collections were made from both of these clusters of populations. And when the collections were made, there was good numbers of plants to collect from. It was thought there were taxonomic differences between the northern and Wongan populations, and genetic and taxonomic work has shown that the northern populations are actually a different species. In the intervening period, populations were found south of Wongan Hills. These were found to be a separate subspecies, and seed collections have now been made. Since the Centre's beginnings, the collections have been actively used for recovery actions such as plant translocations. The results from these translocations are helping refine estimates of how much seed we're likely to need to get a given number of mature plants in the field. Here are the results of numerous translocation plantings for 4 of our threatened species. It shows the cumulative proportional losses at each step of the translocation process. If the aim of our translocation is to establish a population of 250 mature plants, then for a species such as Grevillea humifusa, where just over 25% of seed make it to become a mature plant, we would need around 900 seed to achieve this goal. For a species such as Divisia basarioides, where less than 5% of seed produce a mature plant, we would need in the order of 2,500 seed to establish 250 plants. The success of the centre can be seen by its positive contribution to species recovery. There are over 20 species for which populations have become extinct in the wild, where seed collections were made before the populations disappeared, and which are now safely stored in the seed bank. Seed from the centre has also been used to translocate over 50 species back into the wild. Thank you.
Good morning, ladies and gentlemen. My name is Ronnie. Today I will present seed exploration and collection of wetland riparian in the downstream of Brantas River, East Java. Introduction. Height of biodiversity in Indonesia depend on wetland riparian area, especially in watershed. One of the important watershed of the people of East Java is the Brantas River Basin. Unfortunately, its sustainability and biodiversity is threatened by area degradation and increasing river pollution. Thus, the inventory and documentary of its plant diversity is important. This is a map of Brantas River in the East Java, Java in here in the Indonesia. Botanic Botanic Garden has a responsibility to conserve plants, especially in the lowland area, with the main purpose of conservation, research, education, ecotourism, and environmental services. One of the conservation efforts conducted by Botanic Botanic Garden was the exploration and the collection of seed of plant species along the downstream Brantas River Basin. This is the area of downstream Brantas River. Methodology. The study used survey method from 17 to 28 November 2020, and to determine which plant to harvest, seed cut test was implemented. The fruit were extracted, the seed obtained were dry, and their moisture content were determined using the second method. This is the time schedule. Research and discussion. This is the seed exploration and collection diversity. This is the list of seed collection from the exploration. This is the habitus and the seed character. The conclusion: out of 50 species obtained, most of them have orthodox seed, such as Acacia verniciana, Acanthus ilisifolius, Coix laxima-jobi, Cortalia juncea, Sciatula prostrata, Dolichondrone spatacea, Ipome carnea, Lamnea mandalica, Neptunia plena, Loelia tuberosa, Sena hirsuta, Sena sp, Sonaria cassiolaris, and Thespesia purpurea. And only one of them, Sesuvium portula castrum, has recalcitrant seed. Acknowledgement. We would grateful for the Royal Botanic Garden Kew UK for grant award by Threatened Biodiversity Hotspot Program Indonesia. That's all my presentation. Thank you very much for your attention.
Thanks very much to all of the speakers, and thanks out there in TV land for bearing with us through those technical difficulties there. We do have time for a few quick questions. Let me just see here. Where are we? The first question is one for Deanne. It's from Ella Plumans, who says, who asks, what do you see as some of the challenges in seed banking to getting towards collecting those 97% of species that still remain to be collected?
I'd like to answer, David.
Yes, thank you.
Yeah, thank you, Ella. Yeah, yes, sure. There are some challenges to collect the remaining target, but yeah, that's what I said in my presentation, that we would like also to extend our collaboration to those of you in this forum. Thank you.
Thank you. There's a question for Jai from Peter Cuneo, and it's actually a question that I had myself. He says, great talk and impressive progress, which I agree. Do you have an ex situ prioritisation framework for threatened species in China? So what would you collect next? And how do you decide what to collect?
Okay, thanks. Actually, the priority was followed the 3E, endemic, endangered, and economically important for as the standards. So the next phase we are thinking of to focus on more on the crop-wide relatives, and that actually because this is the main, because of limited time. Actually, as I mentioned, we also have the tissue bank, and we had a very good team now working on cryo. So for those endangered, threatened species which are potentially produce the recursive seeds and the intermediate seeds, now we use the cryo to working on it. So so I think the next phase, well, we are thinking of to working more on those.
Thank you. There are quite a few questions out there still, but I just wanted to ask Andrew a question. If you could have the resources to do what you really want to do, what would be your main priority there in Western Australia in terms of collecting in the next decade or something like that sort of time frame?
Okay, well, I guess it's a balance between consolidating collections of the species we've already got. Often the genetic diversity of those collections is low, or as I pointed out, the number of seeds that we have is also quite low. So to work towards having enough seed to do the recovery work that we want to do, and then that comes into the prioritization of what we need to collect. Balancing that is also focusing on our priority species. So hopefully at this point in time, they're in a better condition than some of the threatens, and the earlier we can get in, probably the better those collections are, and the diverse, how diverse those collections are. So it's sort of balancing up those things. And again, prioritizing the species that are likely going to need imminent recovery work, and making sure we've got adequate material to implement that work when we need to. Yeah, it's a whole lot of juggling.
Yeah, sure. Ronnie, I wanted to ask you a similar question. Where do your priorities lie for your program? Maybe, did you hear that one, Ronnie? Sorry, just having a bit of trouble hearing you there. No, sorry, I couldn't hear that one. Well, sorry about that. I'll try again in a second. Maybe we are running out of time here a bit. We're actually into extended time, but I hope people will indulge us a bit because we did have a bit of downtime there. It's a question from Tom North for both Deanne and Jai. Are you looking at storing non-seed collections such as spores? Maybe Deanne first, and then.
Yeah, sure. Because we are rich in the flora diversity in Indonesia, and also there are many potentials to learn more, to know more. What's the storage behaviors they are? They may be, we can say recalcitrant, but we need to further research, and maybe it's potential to be intermediate or even orthodox. We don't know yet. So a lot of potential and a lot of more work to do. Thanks.
Thanks. Yeah.
Yeah, thanks, Tom. Yeah, we tried actually before for the spores. Actually, Q Gardens also trying to get involved, invite us to involved on the working on the pollen, cryo pollen. So I think at this moment still we are, because we still didn't have enough people to working on that. So it's quite tricky. So at this moment we are for the ferns, we are mainly through the tissue bank to preserve. And actually for China, and what we learned is it's quite also quite. We tried some like tree ferns. We tried some, but not very successful. Yeah.
Thanks.
Thanks.
All right. One last one. There are a couple of questions I haven't got time to pass on, but it's another one for you, Jai. There's a question here from Mitchell Corder on the number of medicinal plants that you've got. He's interested to hear that there are quite a few. Do you have plans to do our research and development in future on those, or do you just see yourself as being a store for those things?
Yeah, for medicinal plants, we are actually the figure I choose was we could always have the medicinal properties, because the traditional Chinese medicine actually they have a kind of very strict criteria what kind of species should be listed in or could be traded in the market. So for those, we do have some species, medicinal plant species. They have the problem from the seed storage, like Paris, a kind of very important medicine. Now now they harvest from the wild mainly, and the seeds had very long time to germinate, which take sometimes some species will maybe up to 2 years to germinate, and it will take a very long term to grow before to harvest the rhizomes. So actually for the medicinal plants, well, various issues. What we just treat them as orthodox or non-orthodox species at this moment. So, but it still need to be prioritized. Which one need to be studied first?
Yeah.
So yeah.
Well, thanks very much to this set of speakers. They were really good talks, as were the talks in the previous brackets of this session. Really great deal of new and really interesting information there. Thanks, everyone. We'll wrap the session up now. I just want to share my screen here. I've got some housekeeping things. So let me see if I can just do that. And not sure if that's actually working. It should be. It might be because we're still on the split screen. Yeah. Okay, great. Thanks. So, yeah, just a reminder for people there about the socials, which tags to use there. Very important, please join together on the GatherTown platform. The password is germination with a capital G. There is a link on the portal banner, so up the top of your screen there when you first go in. Big thank you to the conference sponsors and partners. These sorts of conferences don't happen without that sort of partnership. A plug for the next theme after lunch, Season Culture and Society, with Catherine Whitehouse chairing that. That should be great. And again, big thanks to all the people behind the scenes that are making this happen, including the two reporters for this session, Mark and Brad. Thanks for beavering away there in the background. And with that, I think we are finished. Sorry for going over time. Have a bit of lunch and please take a look at those posters. Thanks very much and signing off. Bye.